Hola, yo soy Rosana y voy a presentar uno de los trabajos que me he llevado a cabo en el laboratorio. Y voy a hacer mi presentación en inglés, que se titula Towards Sustainable Plant Disease Management, Understanding the Influence of Global Change and Local Environment on Nematode Communities in Mediterranean Olive Groves. So the first thing that I like to highlight is that according to IPCC, global change and desertification is one of the biggest challenges that we will be facing in the future, and this is going to particularly threat agriculture, as it is going to put even more pressure in already degraded environments. Some of the changes that we can expect is the decline in plant production, followed by a decline in soil fertility, at the same time a decline in plant coverage and therefore in species richness. This is particularly concerning in the Mediterranean basin, as it's going to be a highly susceptible place to desertification, but it's also a biodiversity hotspot, and we are primary producers, so agriculture is really important in the area. One of the cultivars that is going to suffer is the olive tree, which is of a huge economical and cultural relevance in the area. At the same time, the olive tree is host to more than 250 nematode species, considering both free-living and plant-parasitic ones. Moreover, up to 10% of production losses can be caused by plant-parasitic nematodes, according to previous studies. Here you can find some of the nematodes, the plant-parasitic nematodes that we find in olive groves, and we classify them in endoparasites or ectoparasites depending on how deep they get into the root, and of course endoparasites are more pathogenic. All of this introduction was what made us wonder how a nematode is going to be affected by an increase in aridity, and if management strategies have any chance cushioning the effect that desertification is going to have on them. For that, we carried a diversity study, and it's important to note that, of course, diversity is not evenly distributed, but that there are biotic and abiotic gradients that modify it. And to study diversity in several dimensions, we use primary three measures, which is alpha diversity, beta diversity, and gamma diversity. And these indexes allow us to spot some important processes, like, for example, biotic homogenization, which is when specialized species or functional groups are replaced by generalist taxa. And we identify it because we can see a decrease in beta diversity, and this can be due to different processes, like, for example, a loss of species, a loss of function, or even just a change in abundance in the community composition. To answer these questions, we did a huge sampling. We included 52 fields across 5 countries. 20 of the fields were organic and 32 were non-organic. The first step was the nematode extraction and identification, which is a key component, because without this step there are no results, even if sometimes it's a bit time consuming due to the existence of cryptic species. So it's really important to do it properly. And we found a huge diversity, both taxonomical and functional. Like, for example, we ended up finding 111 species, and the most abundant and prevalent ones were Paratylencus and Helicotylencus. At the same time, we collected 71 variables that we analyzed and organized in different groups to make it easier to study. Like, we included climatic variables, spatial ones, management, of course, but we went beyond organic and non-organic, and we looked at particularly practices that were being carried in its field. We also look at soil, like, for example, organic matter or caking, but we also collected the residues that were present in the soil, like insecticides, antibiotics, and all of them. And of course, we collected different plant parasitic nematodes diversity variables. We studied species richness, beta diversity, community dissimilarity, and some functional diversity metrics, like for example, the percentage of endoparasites in the sample. So our final goal was to see if this diversity followed any pattern, but of course at first glance we couldn't identify it. We got to dig deeper. And so we took the 71 variables and did a variation partitioning with the goal of seeing if they were able to explain the change in nematode communities. And this is the model that we created. And we saw that it was able to explain over half of the variation, and that this variation was explained mostly by the environment, so the variables that we had named, and that only 6% of this was explained by a space. We spot a lower stochasticity compared to other studies before, which can be due to the larger sampling frame that we had. And so these are the variables that I named before, and this is how we organize them. We saw that the one with the highest impact was climate. It is probably also related to having a larger sampling frame. But we also wanted to see how they were interacting. And for example, some things that we saw is that there was no relationship between climate and management, so that climate does not define a particular management practice. That the biggest interaction was between climate and soil, which confirms this verticality of the system and the top-down control. After the variation partitioning, we confirmed the interactions that we had, and we used that to build a structural equation model, which is going to allow us to look at the direct and indirect relationships within the system. And even though we can be a bit overwhelmed by all of those arrows, I think it's important to focus on the impact that climate is having over nematodes, because it's pretty strong, and it's the relationship that is going to help us answer our first question. And we confirm that increase in aridity is related to a decrease in beta diversity, which means that we are facing biotic homogenization when facing desertification. We looked at the other two indexes, and we saw that this homogenization is not due to a loss of species, but to a decrease in the evenness of the community. And evenness and beta diversity are both related. But of course, this makes us wonder which are the species that are benefiting from this inequality. So we looked at the percentage of endoparasitic individuals, and we also studied the proportion of plant parasitic nematodes when compared to the total abundance. And we saw that this is benefiting the bad guys. So endoparasitic and plant feeding nematodes are the ones that are increasing its presence when we increase the pressure in the environment through aridity. But we also wanted to know if we had any chance to modify this by the management strategies. And sadly, you can see that when we compare organic and non-organic, there were no difference between them, and that biotic homogenization happened anyway. But how is that? Well, if you remember the previous slide, because this relationship simply doesn't exist. So organic and non-organic is not affecting nematode communities. What is affecting them is particular management practices, which gives us a hint of what's going on. And so organic and non-organic is not going to define if we have abiotic homogenization or not, even when we see some differences between both of them when we look at species richness, for example. This makes us think that probably the organic and non-organic classification is not enough to ensure the protection of soil diversity. We probably need to try a little bit harder because, as we've seen, at least what's happening in nematode species is that more diversity means healthier soils and more resilient soils to disease, as more pathogenic species do not have the chance to thrive. And of course, we have lots of plans for the future. For example, looking at how natural environments are behaving, if they're going to have the same response or a different one, to look a little bit more into functional diversity, or to simply explore the other relationships that we saw in the structural equation model. So this was the end of my presentation. This is my beautiful lab, which I'm really grateful for. Muchas gracias por todo. Un abrazo. Os echo de menos. Y muchas gracias. Espero que os haya gustado. Adiós.